Hi everyone! Today's presentation is about DNA and how DNA replicates. This is a pretty big topic in USABO in particular, and knowing it can help you get a lot of questions even if it's not the main point. So it's an important baseline to have so you'll have a better problem-solving experience. So the table of contents for today's presentation is first DNA basics, then DNA replication, then DNA proofreading, and then finally DNA packing in the nucleus. So first off is DNA basics. So to start off, we have DNA experiments. In the beginning, people weren't sure whether DNA or proteins were the genetic material. However, the case for proteins seems stronger because proteins were more familiar to the researchers back then, and DNA just seemed too simple. Since proteins were made out of a bunch of amino acids, and DNA only had 4 bases to work with. So in 1928, a scientist named Griffith started doing some experiments. So he determined that bacteria were being transformed by something. So transformation, it's important to know, is when bacteria share DNA with each other. However, in this case, usually one of the bacteria is dead and the other bacteria is picking up that bacteria's DNA from the environment. And it's also important to know that transduction is what happens when a virus, a bacteriophage, infects a bacteria and then transports that bacteria's DNA into another bacteria. So yeah, he discovered that, there was this something causing bacteria to have changes. And researchers Avery, McCarty, and MacLeod identified that thing as DNA. So in 1952, Hershey and Chase determined that bacteriophages, which were basically DNA plus protein, they're viruses, reprogram bacteria. And they were thinking, hey, this is already a technology that's pretty developed, so why not experiment with it? So their first batch of bacteriophages they made had proteins that were tagged with radioactive sulfur. And their second batch was DNA tagged with radioactive phosphorus. And then what they did was they centrifuged the results after those batches of bacteriophages infected the bacteria. And based on where the radioactivity was after they centrifuged it, it was determined that DNA was being injected into the bacteria, and the protein coats, which were tagged with sulfur, just kind of fell out. So this really confirmed that DNA was the genetic material and not proteins. So in 1950, someone named Chargaff studied DNA and DNA base pairing, and he came up with these rules. So his rules were that the percentage of adenine is equal to the percentage of thymine, and the percentage of cytosine is equal to the percentage of guanine in a species. And also that different species have different DNA composition. So these are 2 important rules for you to know. And then lastly, in determining the structure of DNA, the main people were Watson and Crick, Wilkins, and Franklin. So Franklin used X-ray crystallography to determine the structure of DNA, but Watson and Crick are usually given credit for the first complete structure. Okay, so now is DNA replication. Okay, so we'll start off with experiments here too. So there were 3 models for how DNA replicated before we knew the actual model, and these were conservative, semi-conservative, and dispersive. So the conservative model, if you'll see here, is that the parental strand is completely conserved. So all of the new DNA is completely new, all of the old DNA stays and is completely old. Semi-conservative is when all of the product DNA is half new and half old. So if you see here, the parental strand, half of it goes to each of the new strands. And then lastly, dispersive is that the DNA is just in chunks and it gets roughly half new and half old DNA as well, but it's not like there's a continuous strand of old and a continuous strand of new. It's dispersed, like the name. So there were researchers Messelson and Stahl that came up with a great experiment to determine what model it was. So essentially, what they did was they cultured bacteria in a medium with essentially heavy nitrogen. So all of the DNA they made was heavy. And then they transferred the bacteria to a medium with lighter nitrogen, which is nitrogen 14. And then what they did was they centrifuged the DNA after the bacteria made their first replication with the lighter nitrogen. And then they centrifuged it again after the second replication. And then you can make predictions for all of the models on how the centrifuging would turn out. So if you look at it, the conservative model after the first replication would have one heavy strand and one light strand, right? However, this wasn't what the results looked like. The results looked like this. So we knew right away that the conservative model was not it. So we still have semi-conservative and dispersive going, because what we expect is that there is one group of DNA that is about the same density. Okay, so moving on, their second result was this. So there's one band with lighter DNA, and then there's another band with kind of mixed, like half light, half heavy. And this corresponded to what the semi-conservative model looked like. However, for the dispersive model, you'll see it's a consistent band, and this is not what they got. So they knew dispersive was out, and that semi-conservative was the model. Okay, so some more specifications on DNA replication. So in DNA replication, you have to have an origin. So this is where you start. And the origin usually has a specific nucleotide sequence that, enzymes are looking for. And then for bacteria who have circular chromosomes like this, there's one replication bubble, like that. And for eukaryotes, there's a lot of origins. So they look like this. So this makes it easier, like having a bunch of origins, makes it easier to replicate faster, because if you think about it, you're doing the same amount of replication in each bubble, but you're doing it a bunch of times, and the bubbles can meet each other, and then you'll have replicated your DNA in no time. Okay, and then the replication fork is where the replication's going. So if you look here, this is a replication fork. It looks like a little Y, and then that's just where the DNA is replicating. And these are the replication forks for eukaryotes. Okay, so for replication, there's a couple of proteins that you'll need to know. However, it's probably not going to make too much sense right now what they do, but there is a picture on the next slide that's going to put everything into context. So first, helicase unwinds the two strands of DNA. So, at the replication fork, the DNA is separating, right? The original DNA. So that's what helicase is doing. And we need those strands to stay separate when we're replicating, right? So that's what single-stranded binding proteins do. They prevent DNA from joining together because they bind to the individual strands and make it hard for the DNA to snap back. And then topoisomerase relieves supercoiling. So if you think about, say, when your hair is tangled, or you have string or something that's tangled, when you try to pull it apart and it's already twisted, it's harder to pull it apart because it just gets more twisted. So that's basically what DNA does. And what topoisomerase does is it cuts one strand of DNA, so all of that supercoiling is relieved. Okay, and then primase makes RNA primers on the separated DNA strands. So it essentially makes these short RNA sequences on the DNA for replication to begin there. And then DNA polymerase 3 is the big one. It's the one that adds nucleotides and does the bulk of the replicating. And DNA polymerase 1 replaces those RNA primers made by primase with DNA, because we don't want RNA in our DNA. In fact, RNA is much less stable, so we definitely want to use DNA for long-term storage. And then DNA ligase joins DNA fragments. So as you'll see in a little bit, DNA replication creates some fragments. So what DNA ligase does is seals that. And leading strand and lagging strand are something we're going to get into in just a moment, but there's only one use for primers, for ligase on the leading strand, while there are many uses, like many times DNA ligase is used on the lagging strand. Okay, so here is what I was talking about, the leading and lagging strand. So essentially, DNA polymerase can only add nucleotides on the 3' end. So if you look at it, the leading strand, that works perfect. It's synthesized in the direction the DNA is unzipping. So the DNA is unzipping this way. And because DNA polymerase adds only on the 3' end, it can just go like this, and it's perfect. However, on the lagging strand, adding to the 3' end means it can only go this way, and that's not what we want, because we also want to be going in the direction that the DNA is unzipping. So what the lagging strand does is that it goes backwards, but it keeps on going forwards in, like, it's kind of like backstitching, or say if you're writing a sentence backwards. So if you're saying, I like apples, then this is equivalent to writing apples like in I. So it just keeps on going in the in the 5' to 3' direction, but it only does short segments before it hops ahead and then does another segment. So yeah. Okay, and then here's where everything kind of comes together. So this is helicase, which we're talking about unzipping the DNA. This is topoisomerase, which is relieving a lot of the supercoiling. And then single-stranded binding proteins are keeping the DNA apart. And then here is the DNA polymerase 3. And then don't worry about the sliding clamp. Essentially what it does is it just keeps the polymerase on the DNA, make sure it doesn't fall off. And then you see on the leading strand, it's just going like this. It's doing great. It's doing it continuously. And then on the lagging strand, we see RNA primase making RNA primers, which are represented in green. And then we have DNA polymerase 3 building off of those primers. And you see it's like incontinuous. And then the fragments you get from, primer and then DNA polymerase are called Okazaki fragments. And this is pretty important to note. They're named after the scientists who discovered them. Okay. And then we see here DNA polymerase 1 is replacing these RNA primers in green with DNA, which is red. So we'll be able to have just DNA in our genome. And then lastly, we see ligase is sealing up these gaps between the fragments. And yeah, so overall, replication's going like this. Okay, and next we'll talk about telomeres. So telomeres have pretty important functions. So DNA has telomeres at the ends, and it's basically a bunch of DNA that has the same repeated sequence, and it's TTAGGG in humans. So what it does first is it prevents the DNA from appearing damaged to the cell's defenses, because after being replicated, the ends of DNA are usually staggered. So they look kind of like that. And like the cell sees that and it's like, oh no, the DNA is damaged. We don't want cancer. So it's going to attack the DNA and kill the cell, which is not good. And it also slows down the disappearance of genes, because if you look here, when genes are being replicated, we're actually losing a little bit. So since DNA polymerase 3, DNA polymerase 1 replaces RNA primers, it can't really do that for the last RNA primer on the end, because there's no 3' end right here for it to work off of. So this chunk that was covered by the primer earlier, it doesn't get replaced, and essentially this chunk is just lost. So we want telomeres to act like as a buffer, so we're not losing really important information. And essentially, telomeres, they give our cells a lifespan. So after a certain amount of replications, our cells are like, this DNA is going to get damaged soon if we keep on replicating it, and, the telomeres are getting really short, so we're going to stop replicating to prevent cancer. So, but telomerase is really, really active. So I should mention what telomerase is. So telomerase is the enzyme that helps with lengthening in telomeres, and in humans, it's only really in gametes, because in gametes we want our cells to be, replicating, and like we want them to have long telomeres because that's going to become a new human. However, telomerase, it's not supposed to be active in normal cells, so normal cells can have a normal life cycle, but it's active in a lot of cancer cells, which is what lets cancer divide indefinitely. Okay, so next is proofreading DNA, because DNA replication isn't always perfect, and also, the environment oftentimes can do a lot of damage to DNA, so we want to make sure that our DNA still has the right codes so it doesn't end up with cancer. Okay, so first I'm going to talk about how DNA can have mistakes or issues. So the thing to know is that DNA doesn't actually mess up a lot. Usually in completely replicated DNA, there's only one issue in 10 billion bases. So it's not really that bad when it comes to, replication issues. So here are where issues can come from. The first thing is that they're paired wrong in replication, which is pretty rare. And another thing is that it can be damaged by the environment. So it can be damaged by X-rays, it can be damaged by UV light, and it's important to know that when it's damaged by UV light, it can form something called thymine dimers, which is if you have two thymines next to each other in DNA and it's exposed to UV light, there's a chance that the thymines could stick to each other and make a 2-thymine unit, which is, a dimer. So yeah, and it can also be damaged by chemicals. So yeah, there's a lot of things that could mess with DNA. So we want to be able to, keep the sequence, consistent and accurate. Okay, so here are some repair mechanisms. Mismatch repair is when enzymes switch out the wrong nucleotides for right ones. Say we are supposed to have ATG at a certain point, and we instead have ACG. Then mismatch repair can, swap the T and the C for the right one. And then nucleotide excision repair, or NER, is when nuclease, which is an enzyme, it excises the damage, so it chops out that part, and DNA and polymerase and ligase fill the gap. So the difference between mismatch and nucleotide excision repair is that nucleotide excision repair covers a much larger space, so it can take out an entire sequence. And yeah, and now we're on to DNA packing. Okay, so DNA is usually stored as chromatin. So there's an important distinction to make. So a chromosome is how DNA is during mitosis. So it's all tightly packed, and you can see that, iconic kind of X-like shape. But chromatin is kind of loose, spaghetti-like, and this is how DNA is normally. There's actually an organization to it, but, from the outside it looks pretty, messy and loose. So DNA is actually stored in a way that looks kind of like beads on a string. So essentially, there are these proteins called histones, and they're represented here with these little kind of quarter cylinders, and 8 of these histones come together to make something called a nucleosome, and you also call this a histone octamer. So essentially what DNA does is it wraps around these nucleosomes, and actually I believe it's nucleosomes are when the DNA is wrapped around them. So yeah, it wraps around the histones, and then we get this thing that looks like beads on a string. And then something to note is that euchromatin is the form of chromatin that's looser. There's also a form called heterochromatin that's tighter. So things to know about these two: euchromatin, U stands for true, so just think true chromatin is the loosest type. And also because it's looser, it makes it easier for enzymes that are responsible for making proteins and stuff to access that DNA. So euchromatin usually means more activity because the looser DNA can interact with more things. Okay, so here is kind of what happens during mitosis to DNA. So in prophase, the DNA starts condensing. And then condensin 2 makes the DNA, which is a protein, it makes DNA loops that get, bigger and bigger, and that makes the chromosomes wider and shorter. So you can see it starts making these loops. Okay, and then pro-metaphase, larger loops happen. So condensin 1, um, which is this green one right here, it makes larger loops from the previous loops of DNA. And then it just keeps on looping until there are, bigger and bigger loops. And then in metaphase, the chromatin is fully condensed and it has a lot of loops. So, we get from that one stranded type of, that one strand of DNA to something like this, which is a chromosome. And that makes it easy in mitosis to, separate the DNA. Okay, and that's the end. Thank you so much. And I hope this was helpful. Please leave a like if it was. Leave a comment if you have any questions, and stay tuned for more.